So I'm going to tell you most of the stories I'm going to tell you about are fly related. So yesterday, going over the posters, there's plenty of fly work, so you guys will appreciate this. But there's a little bit of human stuff in the middle. So basically, the thing I'm gonna, or kind of the storytelling I'm gonna try to tell you today is about how genotype-phenotype maps are environment dependent. And this is, of course, not a surprise for everyone, but I will try to tell you a little bit of how we are trying to approach experimentally these type of questions. So I guess for nobody here will be the genotype-phenotype map context anything new. But in general, it's this conceptual framework that really helps us to try to imagine how genetic variation propagates or not through different layers of this map, right? So you can go through the genome, through these little faint layers that could be development, could be gene expression, could be protein amount, right? What we call endophenotypes, until you get to what we call phenotype, which is a higher order phenotype. Could be behavior, could be morphology, could be disease, right? Any phenotype you want to measure, and ultimately we want to imagine how this distribution of phenotypes maps onto fitness landscapes. So most of what we know about this GP map, and when I say GP map, mostly what I'm thinking about is you are able to link your genetic variation to your phenotype. So a lot of the GWAS and QTL part that Bruce was talking about, most of this knowledge comes most of the times from one environment. Right? We think we understand what are the loci, that regular morphology or disease or whatever, in the experiment we run today in these particular conditions. But the question that is relevant for everything we do in evolutionary biology and ecology, when you think about adaptation, is actually asking what happens if I take my same genetic pool. These are not different species, these are not different populations that have been adapted. This is exactly the same genetic pool. Now you put it in a different environment, right? So this, of course, for forever, we know that when you put populations in different environments, the distribution of phenotypic variation changes. And we also know that the way these phenotypes map onto fitness is also different, right? So we know this from ecology, from the first time we did this type of experiments. But something that remains more unexplored is these little arrows here. So what I try to indicate with these arrows is the genetic architecture of any trait under any condition. And the question is, okay, what happens if now I do this exercise of moving my pool of genetic variation to a different E? Do I get the same loci involved in phenotypic variation? Do I get the same with different effects? Do I get new loci? Do I get less loci, right? So this is kind of the questions that are really key if you're trying to imagine what is the actual potential, genetically speaking, of your population when it's going to adapt to a new environment, for example. If you think about helping non-communicable diseases in human population, when you move from a healthy diet to a bad diet, et cetera, et cetera, et cetera. And we haven't been able to answer this very well yet, not because we don't want to, but because it's really hard, right? Like if you remember Bruce's talk before about GWAS, everything is about power, right? You need statistical power to be able to find these little arrows. And now, if it is a challenging experiment in one environment, you need thousands of samples. Now you have to do this twice, or if you're adventurous, 3 or 4 or whatever number of environments you want, right? So it's really, I would say, more of experimentally limited than the question not being sufficiently interesting, for example. So humans, meaning people studying humans, they are really very interested in this type of interactions, and they have this superpower that comes with sample size, right? They have, as we discussed, millions of genomes, but they have the downside is that they have no idea about the environment in which these humans live, right? You ask them self-reported diets, self-reported lifestyles, but at the end, the heterogeneity of environments is so huge that it's really, it's damping the genetic effects, let's say. In model organisms, we have the beautiful opportunity of controlling environments in our labs, but we have the downside that we haven't quite yet reached sample sizes as the human data sets, right? So we are kind of in between. So what we are actually trying to do is, okay, let's try to tackle these two limitations, controlled environment, sequence enough individuals, and for this we use the fruit fly. Good. But something that is important, I think, and this is a little bit more of extensive intro for you guys if you haven't thought about this before, is that normally we are trying to distinguish how much of our phenotypic variation comes from genes, right, comes from genetic effects, and how much comes from everything else, environment, or things that we imagine are non-heritable, right? But what I want to discuss today in particular, from the experimental point of view, is actually whether these genetic effects are modulated by the environment, right? Not if they are different, not if one is more important or less important, but can I have interactions between these two factors, right? So this is in the literature what you will always find as G by E, or genotype by environment interactions. And just to place this in the context of things that we all think about, is the way we think about how environment affects phenotypes is usually framed in the context of phenotypic plasticity. I guess everyone has heard about phenotypic plasticity. In this particular example, we are showing how the average phenotype changes with temperature, right? So you see that flies and butterflies and many insects growing at lower temperatures tend to be bigger, for example. And then we, and by we I mean ecologists most of the time, they try to model these changes in plasticity using reaction norms, right? So you try to understand what happens to your phenotype when you place your populations in different environments. You can have very robust phenotypes that don't change. You can have linear relationships or funky stuff. This is, again, what environment does to your phenotype, right? But to study genotype by environment interactions, what are we missing from all this theory? Yeah, genotypes, right? Like all this is an approximation to what happens to your average phenotype when you perturb the system, right? So at the end, G by E is genetic variation in this little model of reaction norms, right? So how can we see this like more concretely? You can imagine the same example of your phenotype driven, for example, by temperature. These two lines are two genotypes, and these are your two environments, right? So here, you actually shouldn't care about the phenotype because the phenotype reacts exactly in the same way. Doesn't matter which genotype you have, environment 2 is going to make you bigger, for example, right? So this is not what we are interested in. This is just an average change of your population. But then more interesting things can happen, right? You can have this so-called crossing of reaction norms, when if you check your first environment, you have certain relationship between your genotype and your environment 1. But if this same population were to be put in environment 2, now the genetic effects is bringing totally different phenotypic changes. And this one, which is one of my favorites, you can imagine as cryptic genetic variation, is a situation in which if you're lucky enough to measure your phenotype in the right environment, you are going to find genetic effects. If you are unlucky and you measure it in the wrong environment, you won't see anything, right? So for example, let's ignore the environment 1, just focus on these two dots on environment 2. I decided to make my experiment in environment 2. And then I checked the genotypes of my population. I have red and blue. And my conclusion is this genotype has nothing to do with the phenotype, right? Because they all look exactly the same in this environment. But then one of my students says, Luisa, your environment 2 is too boring. Let's try environment 1. And then I measure the same genotypes. And now, voila, you get a really strong association between which genotype you have and how you look. This is what we call cryptic, because here it's kind of hidden. There you cannot see it, and it gets exposed once you go to, I'm calling it the right environment in the sense that it's actually giving you an association.
So this figure makes a key point in that G by E is two different things. So if you look at the leftmost and the center panel, the genetic variation in both environments is the same. In the second panel, the correlations change. In the third panel, the correlations don't change, but the genetic variation shrinks or expands. So G by E is a combination of genetic changes across environments and also switches and correlations over environments. And this panel nicely shows that.
Yeah. We're going to get also to these effects on variance later on in the talk. Good, but this is nothing new. These plant people have shown us this forever. This is not a surprise if we have been paying attention. And we have, because they can do it easily that we can do with other systems, right? So this is, for example, an example from the 80s where we have 7 accessions of this plant, right? Meaning you have 7 genotypes of this particular plant. If you put them at low elevation, this is how they look. So genotype 1 is clearly the winner, right? Like it's the tallest plant. Let's say that's the best thing. But if you were to take the same genotypes and now you put them at median elevation, then your relationship between who is the best and who is the worst totally changes, right? You could assume that this is the true genetic effect if you didn't measure anything else. And the story changes again if you put them at high elevation, right? So this is just a way of knowing G by is there and is everywhere. We just have been having a hard time kind of pinning down which are the loci that are responsible for this. So we know it's there. Can we map it, for example? Okay. So then the questions, kind of the general question we're going to try to approach in this first part is how does the environment modify the genetic effects that regulate phenotypic variation? So for this, as I said, we are using the fruit fly as a model in general to study complex traits. So if you have done your classic genetics courses, most of what we know from flies historically comes from inbred lines. This is something of what Bruce was mentioning too, that is a really, really powerful resource, because when you have inbred lines, means that you can measure again and again and again your phenotype, and you have really good confidence that what you're measuring is true. You cannot do that if I measure a human, right? Like, it's really, really hard. So this has been this basically kickstarted, genomics in flies in terms of genotype-phenotype mapping, because now we have things that we can accurately measure and genomes that we can actually use for mapping, right? But as it was mentioned before, this comes with the downside that when you want to map complex traits, you need a good sample size, right? So other organisms, for example, Arabidopsis or yeast, they have these 1,000 genome projects, right? So they have really a good number of these inbred lines or accessions that you can do mapping on, but we don't have that in flies, because flies they are not inbred in nature, right? Like we like genetic variation as a fly, and you have a lot of problems with your inbreeding flies. You want to inbreed 1,000, but then only 200 survive, because you are forcing these deleterious alleles to be homozygote, et cetera, et cetera, et cetera. So it has worked very well in terms of sample size for other systems. In flies, we are around, I think, 120 something lines, but I'm not sure, something around there. So what we are trying to do, and not only us, but many people, is, okay, how do we try to compensate for the limitations of sample size, right? And the answer is relatively easy. You start working with populations that are outbred, what means your sample size is only limited, but how many things you can sequence, right? We could argue this is a limitation, obviously, for many labs, right? But let's say, generally speaking, is if you have the money, you could do as powerful experiments as you could actually do, escaping the limitations of inbred lines, right? And there are ways in which you can make these outbred populations in a way that you are trying to control as much as you can for the allele frequency distribution. For example, you can take, these are different approaches that different labs take. You can take flies from the wild, bring them to the lab and do your experiments. Or you can take panels of inbred lines that somebody collected, and you can cross them in a way that is called round-robin design, where you make sure that every genome sees the other genome. So you make sure that when you put them in a cage, drift didn't remove 5 of the genomes you thought will be there, right? So basically, you made A with B, B with C, C with blah, 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 in a circle, and then you meet the babies of those again and again and again and again until you end up with a homogeneous population. This does very good things with your allele frequency distributions. You don't have as many rare alleles as you will have if you sample in our population. You make sure as much as you can that the alleles you think are there, et cetera, et cetera. And this is the type of resources we use, and we are taking advantage of Andy Clark's diversity lines. These are inbred lines, panels of inbred lines collected all over the world, and we maintain outbred populations in the lab for the experiments we do. Oh, okay, and this is for you to imagine. So these are the usual vials that we are all work with when you're working with inbred fly lines, and these are the cages in which we keep our flies. So you can see they can be really, really huge. You can have hundreds of thousands of individuals in one cage, and this is just not so nice picture, but it's just to show you, like this is just full of hundreds of thousands of flies in one cage. So these LD blocks, we break them. So we know that they go down to 200 to 300 base pairs because of the round-robin crossing. You actually don't do experiments with these flies until maybe some 80, 100 generations, until the LD has gone down. So let's say you kind of design these mapping populations for them to be really good at mapping. Okay, so the first story I'm gonna tell you is, I'm repeating myself again and again, but how does the environment modify these genetic effects? And of course, to approach this question, you need an environment, right? And this is our favorite environment, which is high sugar diet. You could pick whatever you want, depending on the ecology you want to explain, or depending on how easy it is to make these experiments, right? So high sugar is a really interesting thing because, as surprise, surprise, flies are model organisms to study obesity, for example, and diabetes in humans. Because flies get exactly or very close the same phenotypes and the same disease-like phenotypes that we get when we are diabetic. They get insulin resistant, they get obese. You dissect a high sugar fly and it's literally white, what you get out of the body, right? It's that body of fat body increased a lot, they die faster, they are sick, right? So high sugar is good because we really understand a lot from the, let's say, disease point of view and metabolic point of view, what is this environment doing to your individuals? Plus, the other component that is easy, right? Doing a diet experiment requires that you prepare your food with different components, and then that's it, right? You don't require anything special. So this is just to highlight the point that when you do experiments, is this really fine interplay between what you want to study, which is the question you want to answer, and is it feasible to do it in the lab, right? Which at the end means we have to compromise somewhere, always, right? Good. And for this particular story, our phenotypic variation is lifespan, right? Why is lifespan interesting? You always try to convince people that the phenotype you chose actually means something, right? We like lifespan because lifespan is this really complex phenotype that encompasses everything, right? If you have a good immune system versus a bad immune system, lifespan is penalized. If you are too aggressive versus not so aggressive, if you mate a lot, if you don't mate a lot, all this stuff, if you fly too much, you are too energetic, or if you are a fly that stays on the bottom of the cage, right? Like all these things, our lifespan at the end is this, let's call it, global representation of what's going on in the organism, which is a cool phenotype to study. It's also a good proxy for fitness in flies. This is not true for every organism, but flies are laying eggs every single day of their life, right? So this is a good proxy of your fecundity, of your number of babies, and we could argue the details, but let's say it's as good as it gets a proxy of how many babies you have without having to measure it. And the last part is that these two things are really highly connected. You can manipulate lifespan by manipulating diet. And I'm pretty sure, I don't know who is here interested in living forever, but now everyone is like, oh my God, let's reduce calories because that extends lifespan. And this is in worms, this is in mice, this is in flies, this is in humans. It's kind of a universal relationship, which is cool experimentally because it means it really allows you to manipulate your phenotype, right? Okay, so the question is easy. Let's identify the diet-dependent genetic basis of lifespan. So how did we do this? So we took a population from Princeton. This is, again, one of these huge cages, hundreds of thousands of flies living, let's assume, happily in these cages, right? Having normal interactions, right? They are flying, right? We are not keeping them, for example, in a tube. They can move, they can interact with different flies, they can mate randomly. And then we said, okay, now let's split this population into our replicates, and we put the lucky ones in the control cages. They are eating the normal food that they eat, for forever. And then we put the unlucky ones in high sugar stress, right? So basically, just media that has 22% versus 7% sugar as the control. And then, this sounds funny, but literally we did nothing else but wait until these flies became old, right? Remember, we're trying to study lifespan. So what we did is we took first a sample of flies from these populations to estimate allele frequencies at time 0. And then we let these flies, and I mean it, we just let them age and die until we found around 500 flies left in each cage, and then we stopped the experiment, right? So when the high sugar flies reached 500 population size, they were around a month old. When the control flies reach old age, they were around 2 months old. So this is just the first approximation that diet is actually not fun for flies to live in, okay? And then what we did is we collected all these last flies, again, to sequence them and estimate allele frequencies at time n, so at the end of the experiment for control conditions and at the end of the experiment for high sugar conditions. This is important to mention that here what we did, actually we did a lot of work, okay? I don't mean we literally waited for them to die, because what is important here is that you don't have overlapping generations, right? You need to be sure that the population you are measuring when they are young is the same population you are measuring when they are old. You cannot have babies coming in because then, of course, then the experiment is a failure, right? So what we did, it was change food every 3 days, I think. All the time we were giving them a fresh food, which means all the eggs that they were laying on that food were coming out of the cage, right? So we make sure that there's no new babies coming into the population. Okay, so what is the idea behind this experimental design, right? So the assumption is that lifespan alleles will change frequency over time, right? And this is just a toy example. Imagine that you measure the allele frequencies at this particular SNP when the population is young, and young means 2 days old. The frequency of this allele is 50-50. You have 3 and 3 flies from this particular allele, for example. And then you measure the same population when they are old, and now you measure the allele frequencies again, and it looks like the blue allele decreasing frequency and like the gray allele increasing frequency. Of course, it's all relative. You don't know what happened in the middle. This is the black box of this experiment. I cannot tell you why these little flies die. It could be that they were fighting too much. It could be that they were sick. All the reasons I gave before for why you could die. But what matters is that these guys with the C allele, for whatever reason unknown to us, are less fit and they die faster, right? So this is kind of the assumption. Things that will kill you will disappear from the population or will reduce frequency in the population, and things that will make you live longer will increase in frequency. So we sequenced a bunch of flies. These are not all the flies that I'm going to talk about today because we did other bunch of experiments to tackle the theory of aging hypothesis. And then we individually sequenced them. So they are barcoded for each individual fly. And we took a low coverage approach of 1x. This is kind of trying to approximate the parameters that POLYGENIC has determined that is a good sampling of your individual flies. And we end up with around 270,000 SNPs. As was mentioned before, this is for trying to identify genotype to phenotype association, right? So you clear out everything that is rare just because you have no power to detect stuff like that, and you prune LD for many reasons, including statistical power when you are doing tests, with the downside of losing these linkage blocks. But again, as I mentioned when Bruce asked, the LD of these populations really decays really fast, right? So this does a good coverage of the Drosophila genome, which is 140 or so megabases. And then, because these are low coverage, we cannot use genotypes, right? We cannot assign genotypes like if you will do in a GWAS. So we model our read counts using beta binomial regression frameworks. These are things that people use when they're using allele-specific expression, for example, which is basically you are modeling how many reads you get on the reference allele over total number of reads, for example. So this allows you to use all the data that you collected while accounting for which individual is it coming from. And the question is, does allele frequency change with age? Okay, so this is what I want to highlight here is a traditional GWAS. You have a phenotype that you are trying to model given their genotype, right? In our approach, we are using allele frequency at this particular to be modeled given age, that in our case is a time point. I would like to ask you guys, what do you think will be really tricky if we were using a traditional GWAS approach? Think experimentally. This same design, exactly what we did, but now we want to use this type of models. Luis? Not quite, not quite. Let's think about this. If I need a phenotype, what does that mean? It's what? Not quite, not quite. Think about it. You need a phenotype of age to death for 10,000 flies. What does that mean? You need to measure the phenotype, right? So you need to take individual flies. You cannot put them in a cage, right? Because you wouldn't know, right? You have to have them in individual things, have somebody scoring them every day, 10,000. I don't know how 10,000 look, but that's a lot. You have to score them until they die, right? Because you need the individual phenotype of this particular fly. With the downside that if they are housed alone, then maybe the social interactions are not as natural as they will have. They probably won't fly, et cetera, et cetera. So this will be tricky, doable. People do insane experiments, right? Like you could do it, but it will be tricky. And the second thing is the genotype. What happens when you are running this type of models? If you have a phenotype for a fly, it means that the genotype, I have to be able to call the genotype in the particular fly, right? And I cannot call genotype with 1x, right? 1x is like one read of this. You don't know. Do you see where I'm going with this? If you want 10,000 individuals, you need 10,000 phenotypes and 10,000 genotypes. So this is probably, I don't know, we could argue, but it becomes probably a non-possible experiment. If you want to sequence, let's say on the lower side, 5x coverage for every single fly to have confidence on calling genotypes, right? So this is just to tell you that we were very clever with this approach. This is what I'm trying to convince you. Of course, it has downsides, and we can argue that, right? But doing this little trick, you actually don't measure the phenotype. You actually don't need a genotype because you are modeling the gene counts. Okay, if you had like the whole population, for example, dying super quick, and then you have less samples. Yeah, I wouldn't know if that will matter for the association results. I will maybe argue that if you have the individual level phenotype, then you gain much more information. Because now here you're saying young and old, right? So I can tell you young and old, but if you had the individual phenotype, then you actually measure between individual variation in aging, which I think, to me, ideal, but prohibitive experimentally. That data. Within generation. Yeah, exactly. Sorry. Go ahead. So the young flies are 2 to 3 days old. So you let them close and mate. You don't want virgins and any weird stuff there. So this is the first sample. And then when they died, meaning at 1 month in high sugar conditions and at roughly 2 months for the control. 1 month and 2 months. In dying? Within a population? Yeah. It doesn't really matter. If you have the ones dying first, you can assume under this idea that they are dying because they have a bad allele, right? For example. Therefore, they are cleared out of the population faster. So at the end, when you're going to check the frequencies, you are not going to see this particular allele. So it doesn't really matter that they are This is part of what you are able to measure, right? That the ones that are dying faster, for genetic reasons, we will assume. But it's the variation that is giving you the results, right? So if you didn't have differential mortality given genotypes, you wouldn't see anything. So your allele frequencies at the beginning and at the end will be the same. You need them to die, and they need to die differentially. If they don't die differentially, so if there's no variation in dying time, in death time, or whatever you call that, then there's no point, right? Like you start from the assumption, no, this is quantified, that there's a distribution of time to death in a population.
Yeah, that's why when you use inbred lines, you can measure the average lifespan with incredible precision. It's these outbred populations where, a fly could die because it got squished when you put the cap back on wrong. My quick comment was that your approach is a very nice one. It shows that interesting ideas are independently reinvented. So in animal breeding, this is called birth date selection mapping, BDSM, which they have interesting t-shirts for.
Date selection. Yeah. And humans, actually, this project was inspired because in humans, they actually tried to do it. Because, right, this is actually a very interesting design, right? So you have the UK biobank. You have thousands of people, thousands of genomes. Of course, they are most of not dead, right? So what did they do? They took the genotypes of these, whatever, 100,000 people, and they asked them, when did your parents die? So this is kind of a proxy, right, of time to death, because you cannot do better. And they try to run this type of approaches to try to see, is there any association between genotype and when our parents die, for example. And this was kind of the inspiration, because we can do it experimentally, right? They cannot do it experimentally.
Yeah, they've also met with cohorts that are 90 and older.
Exactly, yeah, right. Yeah. They have found super interesting stuff in these long-lived studies, yeah. Good. So at the end, what we are looking is, if you want to visualize it this way, is patterns of allele frequency changes that are shared between diets, and patterns of allele frequency changes that are not shared between diets. And that's what we are trying to call shared genetic effects. I want to be dramatic in my statement, so you can think about it as universal aging loci, right? It doesn't matter where you place your population, they're always there, or things that are diet specific. So just a quick summary of the results. At the end, our main conclusion is that high sugar unmasks cryptic genetic variation for lifespan. And this is, again, you're probably going to see this again and again and again in this course. This is a Manhattan plot for our association. This is the p-value and the location in the chromosome for every single SNP that we tested. Everything above this line was significant. We use a permutation-derived approach, as Bruce was mentioning in his course, to derive these particular significant thresholds. And you can see, if you're paying attention to the details, that the thresholds are different between diet. And this is another message that I want to tell you, is this idea of deriving your permutation, your significant test from the behavior of your data is gold, right? Like this is, because we assume a lot of things when we are taking a Bonferroni approach or anything that assumes a distribution of results, but actually real data is funkier, right? Like there are a lot of things that are going on in your data that you have no idea. Nobody thought about anything that could match the distribution of weirdness. But if you are deriving these thresholds from permutation, you don't need to know what is weird, but you only know that you are accounting for it, right? Like whatever weird behaviors you have, you are going to catch them when you are doing permutation-derived p-value approaches. Good. So just a way of summarizing this thing, just to bring the point of the cryptic genetic variation, is that if these were all the loci we found, 60% of them, the gray dots, 70% of them, sorry, are shared. These are my universal lifespan alleles, let's say. But we find this really interesting number, which is 30% of loci that we were only able to map in one condition, in this case, in the stressful condition that is high sugar. So this is important because cryptic genetic variation is this really interesting parameter that people studying evolutionary biology really like to consider when we are thinking about why, for example, is genetic variation maintained in populations, right? We have all the reasons that we can imagine. One of those is that you have GY interactions. You have environment-dependent genetic effects, which means selection can only see you if you are in this particular environment, but then you change to the other environment, and now the genetic effects that are relevant there are different, and selection cannot see these ones, right? So this is one of the reasons why we think this is important. Another reason is evolvability. So I know a lot of people here think about that stuff, is if you have this hidden genetic variation there, and now I put you in a new environment, Now you have these new superpowers you didn't know you had, right? That were there, just probably neutrally accumulating, we don't know, right? But they are there. And they are like these magic cards that you can use when you are coming to a new environment to try to adapt, for example. They could be detrimental, sure, right? But there's also the potential that this hidden variation that is there is going to be potentially advantageous in a new environment. But again, it's really hard to put numbers on this, right? So I keep bringing, like, a lot of the things that we think are interesting in biology is sometimes really hard to put numbers on it because it's all about statistics, right? It's, can we actually see it? And this goes down to, are we able to map it, right? So this was very interesting because then we know there are tons of genetic effects that are unmasked under certain conditions. Okay, this is just, if you're trying to imagine how does it look, because for us experimentalists, we have to see it. Yes, yes, yes. Oh yeah, oh, sex there was fascinating. So these populations are male and females, because again, we are trying to preserve, you know, like real population. But the patterns of aging, of course, we know that from flies, is different between sexes, right? So you have in some cases males, or depending on the diet, I can tell you details about it, because we found really striking differences in age given diet, which is also super cool. So all that is there, and we have some analysis for the sex part. But this is a good point that I wanted to mention. It's good that you mentioned that, is when you run real experiments in this sense, that you have a lot of things that could potentially confound your result, you have to account for them, right? So for example, we got plenty of beautiful results when we accidentally forgot to account for sex differences. And then we were like, oh, wait a second, actually they are dying differentially. So the models that we ran when we were able to account for this, it was actually different from what we thought it was, and then you go and you can track it to artificial results that are inflated by the difference by sex. So just again, right, like running proper models that way you can account for your covariates in this case, it's very important. Good. So this is how it looks. If you imagine these are your T0 samples, a shared genetic effect is an allele that increases in control cages when they are old and increases in high sugar cages when they are old. This is just a little visual example for you to see it. A cryptic or environment-dependent effect are allele frequencies that didn't change in control, for example, compared to T0, but they decrease in high sugar, right? So this is actual data of how these allele frequencies change. So this, I bring it here because then, of course, we have been talking about allele frequency spectrums and what does it mean, and where should alleles that are selected and not selected fall in this distribution. So what you can see here in gray is our so this is the allele frequency distribution of this particular population, as all populations is inflated towards rare alleles. And these beautiful and interesting lines are the allele frequency distributions of the loci we're able to map, right? So you can see how different they are. Average 0.3 frequency of these particular alleles, right? So my first message, and we can talk about it, let me know what you think, is power, right? Like this, we all know when we are doing mapping, the effect sizes of complex traits, and lifespan is certainly a complex trait that probably have a huge tail of really, really small effects. Even with this sample size, we cannot really capture things that are rare. And this is not even getting into the super rare, right? But still, it's really hard to get there. But the other one, from a biological point of view, that I find very interesting, is that these alleles are not rare. And then we can come back to the discussion of why do we have genetic variation for fitness traits? This is you know, a whole field of research. Why? Why is this stuff there? And this is super interesting to me because it means there are tons, probably not all, of things that are not on the way out or on the way in of these populations. They are there at high frequencies. Good. And this one is about effect sizes. This is, again, I think it's very interesting to think from you guys thinking about it from the theoretical perspective of what is possible. We have had with a couple of you conversations about this. Is what I'm measuring, as Luis said, like it's within generation selection, right? Like it's what is killing you or letting you live longer. And this is the allele frequency changes for these loci. And you can see they are not small, right? That you have in average, I think, 7% allele frequency change between a population that is 2 days old and the population that is almost dying, which is super strong, right? Like this is a huge allele frequency changes.
These are the alleles that are biggest change.
Yeah, exactly. And this is, for example, the null of when you take things that are not significantly associated. They are, of course, around 0 in terms of effect size. So we always compare to what are the things that we are not able to map and what are the things that are the biggest effect that we map. And the interesting thing here, too, is that this line is the effect size in control conditions, and these two are the effect sizes in high sugar conditions. So this also tells you that it's not only that you unmasked effects, but also the effect sizes are actually enhanced or not, or reduced, depending on the environmental condition in which you are. Right? Okay, questions about this? Yes? Count of the alleles in the genome. Across the whole genome. Yes. Mm-hmm. So basically every SNP. So how many SNPs have this effect size? Mm-hmm. Sorry? No, it's literally a histogram. It's just a histogram, so these are counts.
Yeah, sorry, I'm
yes.
confused by the unit. So this is density is number of SNPs.
It's just a histogram. So you take every SNP and you say, what is the allele frequency change of this SNP? And then for most of the SNPs in the genome is 0, because they are not associated with the phenotype. That's the gray line. And then you take the significant ones and you say, okay, this SNP that I map as significant, what is the allele frequency change? And it's, for example, 0.10. And how many of those are 0.10? Kind of.
Okay, so how many alleles are there?
So this is 300,000.
Between 5 and 10%.
Yes. So the gray line, for example, is 3,000 SNPs in the genome. The gray null distribution of no effects.
3,000.
Correct.
So
300,000. And then the blue one will be, I don't remember the numbers I showed you, 1,000, for example. And the orange one, don't remember now, 700 or something.
So why, I don't understand even in these conditions, why there aren't a lot of 0? Why you have a peak?
At 0?
No, why in later in these conditions you have a peak out of 10.
Exactly. So that's the average allele frequency change in all the loci that are significantly associated with allele frequency.
Oh, okay. So it's the way you filter, it's the way that's filtered on significance.
Correct. Which is associated with the effect size.
Okay. So actually, so there's some distribution if you're not filtering so much significance, which is coming way up and it's a peak of 0.
It's going to be correct.
Yeah.
Exactly.
Okay.
Exactly. Exactly. First, and then I come back. Mm-hmm. I think the mapping was done on a reference and alternative allele. Yeah. So so then probably we polarized by the allele that was. No, I think it was alternate and derived. Yeah. It's a trend. Yeah, I will show you a little bit about the effect of derived and ancestral allele and alternative alleles in causing the effects. But yeah, it suggests that it's one type of allele that is being beneficial. So it's only the derived allele that tends to be beneficial. Could be. Now, I will have to go and check if this is the absolute. It must be the absolute change. Yeah. But I can change the data. That's a good point. Yeah. Remind me, and I can go check. Yeah. Yeah, because if we are looking for what is the average change, then of course it has to be the absolute. Yeah. But I can check the details and confirm that. Yes. Yeah, so we didn't do a deep exploration on LD, but what we tried to do from the beginning was to filter everything that was tightly linked. So we LD pruned, and this is again approaches depending on what you want to increase, or more power or more detection of LD blocks. So we decided to prune them. Exactly. So we prune them. All our SNPs are pruned. You don't have strong LD block. I think it's 0.7 or 0.8 or something linkage around them for, I don't remember. So they are in theory tagging this region, but we don't have the information about the ones. which I like when people actually do mapping that include everything. I really like it because you can see that this really, for example, nice peak decays in association. But again, so compromises and powers.
And you're probably going to generate
that you can, yeah. This is actually will be a very interesting thing to see here because all these things have to reshuffle when you go to a T0 again. Right? All these flies had babies and then the story starts again. So these dynamics of how you, for example, create LD and break it when you have the new population mixing up or stuff like that. Yes.
Larger minor allele frequency changes for the significant SNPs that could potentially either be because, on average, you start with higher minor allele frequencies at T0, or it could be that they increase more than you would expect under just drift, say.
Yeah.
So can one disentangle between
Yeah,
those
so we
two
did a lot of
possibilities?
simulations to try to understand what was going on, changing all those parameters. Like if you start with allele frequencies that are, for example, too low or too in the middle, or actually cover the range of what we know exists in this population, what is your power to detect? So all this is included in the, we can talk about that, like it's long supplementary material, trying to actually figure out what are the reasons of which these things change. A lot of that is you start with higher allele frequencies, and I think it's just hard to detect, for example, if you have very low allele frequencies, changes around that in one direction, for example, right, you're kind of stuck there. And a lot of that is alleles do change also. So you have like a mixture of the two scenarios happening there. But you definitely have differential power depending on your starting frequency. Okay, good. So now I'm gonna tell you this little story that I really, really like. And it's about the evolutionary history of lifespan-associated alleles. So again, people studying humans, they are very interested in this idea of the evolutionary mismatch hypothesis. Who has heard about this? Okay, so the story goes that we evolved, our ancestors evolved in this particular niche, in this particular ecological context, and our genomes had all this time to evolve with our ancestors here. So there's kind of a match between these ecological conditions and the genetic variation that we needed to survive in these conditions, right? So that's kind of the story. And then in the last 200 years, we got here. Don't deny it, this is what we all do. We sat in the couch, we watch Netflix, and we eat not so healthy food, right? So this happened really, really, really recently. So the idea behind this hypothesis is that now you got a super quick change in your environment. So there's a super quick mismatch in what your genome thinks should be doing. Sorry for this wording, and what your ecological needs are, for example, right? So this has been in the literature for a long time, right? We have this evolutionary history mismatch between what our genomes are good for and what we need them to be good for now, right? And they have been using this a lot to try to explain this huge incidence of non-communicable diseases after the Industrial Revolution, right? Like, why are we getting all these metabolic, mental disorders, all this stuff that we cannot explain? They are really complex diseases. What is going on, right? So this is a really strong hypothesis. Unfortunately, for people studying humans, we don't have really a catalog of fitness-associated loci to test this hypothesis, right? Like, to be able to test this, you need to start with, I do know which alleles actually influence fitness now, so I can try to figure out what to do with that data, right? So they don't have it. We are lucky because we just got it, right? So we did this huge experiment, and we are able to map these 2,000 positions in the genome that, in the fly at least, affect fitness, okay? And I'm bringing this here, and I think I had put a little star exactly there, just to bring back the discussion to the point of why do we sequence, right? Like, so there's always this, we know so much from Jain, as we have seen in other lectures, right? A lot of people, when I present, are always, but why do you need to sequence so much, right? You can take another approximations to questions. But I'm just bringing this little story to illustrate that sometimes you need the identity of particular positions in the genome to be able to tackle questions like this, right? So for example, this particular theory has two statements or two assumptions for that to be true. And the first one is that the detrimental alleles today, right, so the things that make us sick today, are most likely evolutionary recent. So they tend to be derived instead of ancestral. And this is just the idea that these are SNPs that help us adapt to these previous conditions, right? So detrimental today are most likely evolutionary reasons. And the second one is that detrimental alleles today were beneficial or neutral at some point in our history, therefore they haven't been purged or disappeared or fixed or anything. So these are the two premises of this hypothesis. You can imagine that like this. So if this is the fitness of the derived allele in an ancestral environment, they were better, neutral, or positively selected. So they were good or neutral, but now you place your population in a new environment and then the fitness, it goes down. This is kind of the framework, right? So then how do we test this? We try to test these two things, right? So the first one is, what is the state of our alleles? They are derived or they are ancestral. For this, we took a phylogenetic approach. So we work with Drosophila melanogaster. Sorry if I didn't mention the species before. With Drosophila melanogaster, and we did is we took data from Yakuba and simulants, which are around 80 million years old, separated in the phylogeny, and then we just scored whether we found the allele in the 3 species, we call it ancestral. If we only found them in D. melanogaster, we call them derived, right? So in this way, we can actually say our allele that increases or decreases lifespan, is it ancestral, is it derived?
How many genes do these have? Some of them have, the other ones don't. The walls are
Oh, of the actual gene? No, yeah, most of them. So the hardest thing here is the alignment. So if you want to align things that are not genic, so that, for example, reduces our data set of, I don't remember now, let's say 3,000 SNPs to maybe 1,000, that you can actually score confidently across all the species. So it reduces your sample size a lot because you cannot align.
Different questions. Whether the differences between them might be as much associated with extra genes that some of them have acquired, the other ones didn't, or they lost in the end.
Yeah, yeah.
So what's the ballpark of that number? Or how many genes?
Overlap between them? Oof,
Yeah. How many don't overlap?
I actually don't know. Fly people, Christian. I will imagine it's most. Yeah, I actually don't have a number for that, but I could check of how many.
No, because I guess what I'm wondering is, my guess, quite a lot of the selection between them
Happens across species.
those genes that don't have it.
That are different, yeah. Yeah, so here we're thinking about it more than in terms of selection, in terms of can I polarize my SNP, right? So can I polarize it by derived or ancestral, regardless of, for example, whether any forces are acting across species, right? It's more literally just to polarize it. Correct. Exactly. They remain there.
They didn't get slaved in the lab as much.
Oh, that's a good point. Okay, so this is again our null, right? If you take the proportion, you take all the SNPs in the genome that you can actually find in these 3 species, right? So you can polarize them. What is the proportion of the longevity decreasing, so the bad allele that is derived, right? So it's 50-50 for the whole genome, right? As it should be, there shouldn't be any reason why these things are derived or not, because they actually don't affect the phenotype. But when you estimate for your significant SNP, so the things you can map, they are in excess of derived alleles being decreasing, right? So this is our test of this particular hypothesis, and the answer is yes, the bad alleles today tend to be recent. The second one is that the detrimental alleles were beneficial or neutral in an ancestral environment. And here we get into tricky territory, because how do you test that, right? Like, how do you know what we don't even know what these things do today, right? How do we know what these things did before? So our approximation to this, which is the best idea we could come up with to test this, is what Bruce just mentioned now, is flies come from Africa around, I don't know, 10K or so years ago. So we know ancestral populations were there, and they migrated all over, including to the US, where our population comes from. So we thought, if we compare allele frequencies of our bad alleles today, and then we go and compare allele frequencies in an ancestral population, maybe the differences in allele frequencies might give us some information, right? You could assume that if you are a really, really bad allele, you will have slightly lower allele frequencies compared to the whole genome, right, neutral evolving. If you are slightly good, you might have higher allele frequencies, that kind of the average in the genome, right? So that's what we did. This is the allele frequency of alleles of things that you can map, right? So these are things we find in the mapping in the normal Princeton populations. And this is the relationship of allele frequencies. We're not making a lot of claims about that, but the bad alleles are lower in frequency than the good alleles. But the interesting question here is not that, but what happens if you go to Africa, right? So if you take African populations, first you estimate what is the allele frequency distribution of loci that are not significant in Princeton, right? That these are like kind of random loci in the genome. And then you take the decreasing and increasing alleles in Princeton and you estimate the frequencies in Africa, right? So these kind of control groups are actually matching all the properties of these alleles in Princeton. So you are not introducing any bias because you are taking a really particular weird sample of SNPs. So they are actually matching the allele frequencies, the properties of these particular alleles. And then what you can see in Africa is that the decreasing alleles, so the bad alleles, the alleles that kill you, are actually slightly higher in frequency than the genome in African populations. And then for us, this suggests that they might at least be not detrimental, right? They at least might have been either neutrally or slightly beneficial. Do we know what they do there? No, right? This is just an approximation to the question based on allele frequencies. Correct. You have to assume that. Yeah. Mm-hmm. So the answer to the second question is yes, detrimental alleles seem to be beneficial, if not neutral, in the previous population. So this is for us to say, yeah, we believe this idea of the evolutionary mismatch might be actually a true explanation for mismatches between the genome and different environmental conditions. Good. Questions about this? This took me longer than I thought, and I still want to tell you a lot of fun stuff. So don't ask me questions, and I get going. Good. Okay, so I told you before that we are trying to figure out what happened with these arrows, right? What happens? How does the environment modify the genetic architecture of these traits? And now I'm going one step higher in this GP map and asking the easy question of how does environment modify the phenotype? We have measured this in every single species that we want. But now I wanna call the phenotype something else. So we had this discussion already, and I hope we keep having it. What is a phenotype? Right? Like this, we could have the whole meeting about that. What is the phenotype? How do we define it? That doesn't make sense. So most of the traits that we studied are very continuously, and most of what I told you now, and most of what I've done, and many people have done, is focus on the mean, right? Like how does my mean trait changes? Remember when I showed you plasticity? What is the average change in my population when I put the population in a different environment? But we cannot forget that these beautiful distributions have variance, right? So we focus on the mean, but the variance is actually a property on its own of all these phenotypic distributions that we study. So you can imagine that we can tell different stories if my phenotypic distribution looks like this versus if my phenotypic distribution looks like that. And studying variance is another word for studying phenotypic robustness. For people who study plasticity, is another word for studying, for example, plasticity. This has many flavors and many noise. A lot of people like to say that we are studying noise when we're studying variance around our means. All these flavors apply. And the question for us, that's what we are putting a lot of our efforts in the lab, is trying to figure out whether this variance as a trait can evolve. Does it have genetic basis? Does it help adaptation? Does it not help adaptation? All that from an experimental point of view, right? A lot of this stuff has been done theoretically before. Okay, this is just if you were not following. When you think about mean changes, is your environment changes your phenotype mean? But there are beautiful also possibilities when your environment doesn't change your optimal, if you want to assume your average phenotype, but it actually changes variance. This is what we're trying to figure out. So the question here is, how does the environment modify the phenotype? Our environment, again, is our favorite high sugar diet. But our phenotype this time, going back, I'm glad I can link to what Bruce was mentioning of how people are studying gene expression as a trait now. Our phenotype is gene expression, right? It's how much reads do you get from one gene in a population. We don't do allele specific expression. Okay, but before you start asking these questions, you should ask yourself a different question before.
Actually, just a quick question.
Yes.
So in your high sugar flies, you see higher fluctuating symmetry?
I'm coming to that. Yeah. So the question before we get into measuring stuff is expression variance. I like robustness because it's a really nice word, so we'll keep using probably robustness. Evolutionary relevant. You will tell me, of course, Luisa, sure. Somebody will say that probably no, this is just measurement error, right? Like it could be noise, it could be whatever. So before you start doing, crazy experiments, you should convince yourself that what you're measuring is relevant. So this was the first question we asked ourselves. Okay, how can we make sure that this trait defined as variance is actually meaningful? So we went back to transcription variance, so gene expression variance, and we asked, is it a gene property? So we are not the first ones to wonder about this. People have done that in some yeast and some cell lines in mice or something like that. So there's not so much, but there's some information. And we thought, we are gonna put ourselves in the worst possible situation, right? I can run an experiment, I can measure gene expression, and I can tell you which genes have higher or lower expression, but that might be experiment specific, right? Like variance could be noise, let's say. So what is the scenario in which which is the worst possible scenario? Is if you take experiments that have been done in different labs by different people, when people were happy, when people were sad, you can add all the noise you can imagine, and you still ask, do I find a pattern in the middle of all this noise, right? So don't worry if this doesn't make sense, I'll walk you through it. So the assumptions here are the following. This is gene A and gene A in two different contexts. You can imagine two different labs, or you can imagine your technician ran the experiment where she was happy and where she was not happy, or your student analyzed the data where we were happier. All this matters. We know this matters. If you ever work with gene expression data, you know what I'm talking about, right? And then you can ask, this is gene expression. Is the variance of this gene A in context A the same as the variance of this gene in lab 2, for example? Maybe it's easier. Lab 1, lab 2, run different experiments. So what you do is you estimate the variance for every single gene. This is the variance in lab 2. This is the variance in lab 1. And every single dot is a gene. If this is true, gene A is going to have low variance in both labs. And gene whatever is going to have high variance in both labs. So you have a good correlation of variance across contexts, right? Across labs.
What's the covariance?
Of across genes? You don't know. That we estimate when we get the data.
So, I mean, large numbers of data sets where two labs are doing the same thing.
The same.
If you do principal components, the first principal component, which lab does the data?
Is lab. Exactly. So let's say we assume the worst. We assume that actually a lot of what you measure is just technical artifacts. In particular in variance, right? That is more prone to this stuff. The second scenario is when actually variance is, for example, induced by the lab who made the experiments, and then you don't have a correlation, right? So gene A is going to have high variance when you measure it in lab 1, and it's going to have low variance when somebody else measures it, right? So these are the two extremes, let's say, of the scenario. So going back to making your life difficult, again, putting ourselves in the worst situation, we went and we screened where the data is available. Data that has been measured in decent enough sample sizes by as many labs as we can, in many countries as we can. What is the most variable data? So this is human. Inevitably, we go down to human data, right? So this is, in terms of gene expression, probably the best organism with available data.
Take one cell type.
So we found 57 studies in humans that cover different tissues.
Yeah.
There's most of this stuff is like tissue specific that have been run in different labs and have been run with different power. And we try to find any pattern that comes out of that.
The tissue dependence is huge.
Yes.
So you're trying to compare tissue.
I'm not going to talk about tissue, but the second part of the project is actually tackling what's the effect of tissue-specific variance. So here we are accounting for all that as if they were covariates in terms of the lab instead of the tissue, we took only healthy, for example, individuals, trying to remove the disease part. So, but all this is accounted in the models, but we also have an explicit tissue-specific variance analysis. I'm not going to talk about that, but it's in the paper. Yes.
say like the ones done at the JNCASR, I mean, they get down to where they claim, I don't know, 2,000 different cell types in the brain, and everything gets subdivided and subdivided. But
Until
you're
one cell.
lumping them together
This is
more.
tissue specific,
Yeah.
Tissue specific. Yeah. Yeah, exactly. So that's why we do experiments with fly after. But yeah, so here you try to remove the environments that are reported. For example, disease or gender or whatever. But everything else that is there that you don't know, it's also contributing to that heterogeneity. Just because the data is there. So for example, if you want to find 57 RNA-seq studies with genome-wide good coverage of expression across labs, for example, it's really hard to find in other organisms.
But the human ones that
you have them. Sorry. That have across different, I don't know, plants maybe? You guys have it? It has it, yeah. As always, we ignore plants, you see. Because in any other, for example, mice has like nothing. Flies, nothing. It's really, really low. And this question was asked before, for example, but in one experiment that was really good covering yeast by one. So I cannot do the trick of, is this variance induced by your experiment, or is it variance induced real, let's say, across different? So actual sample size in RNA-seq now is getting better, but not really. So, but maybe I should actually have thought about plants. Yeah.
But is it the depth on these is typically enough? They claim to be picking up all the mRNAs that are expressed, or is it
yes.
a lot of transcription factors?
They really do good cover data. That's why, and you see the sample? Exactly. So this is like a lot of the stuff here, half maybe is probably GTEC stuff. When they are doing really, really serious coverage stuff. But
tissue-wide, these are not single cell.
No, we haven't gotten into the tissue stuff. Yeah, and single cell is an interesting thing because estimating variance out of this sparse data that comes out of single cell. I don't know. Yeah. Okay. Tissue, yeah, tissue. I'm pretty sure we could do this with I don't know, we'll probably get there, but yeah. Oh, single cell is insane because it's really sparse too. You don't cover the whole genome, you get like, the sampling there is really, really sparse. It's more complicated to handle. Yeah. Okay, so we get 60% of the transcriptome. We are removing the effect of mean. This is important. Every time you talk to somebody working with variance, right? Mean is everything, right? Like there's a relationship between mean and variance in almost every phenotype you measure, specifically in gene expression. So the statistics behind that, the QC, let's say, it has to be very good.
They were very deep. Why only 65%?
Because you are crossing across different tissues, across different labs, across different. So the capture of the genes that are expressed in one tissue are not necessarily the same. Correct. Okay. That you can actually use the 57.
Right. So a lot of them are just sort of general cell.
Correct. So like if you have something really like only expressed in lungs, right, that you wouldn't catch. Yeah. But I think 65, I was surprised, decent overlap given all the changes. And everything we do is rank-based inference because again we can argue what does variance mean, right? What does it mean? What does the scale of variance in this situation mean? Okay. So long story short, this is what we find. So we find that there's this high correlation across labs, across samples, across years in which these experiments were run, and we find very little of low correlation. So this is just literally the distribution of Spearman correlation across studies. So this is literally a pairwise rank correlation between every single one of the 57 studies, right? And you estimate how good the correlation is between all of them, and you see like 70%, 75% of these comparisons are really high, higher than 0.45. If anybody here has worked with gene expression data, I hope you really appreciate how strong these correlations are, right? Like, so gene expression data is extremely noisy phenotype. We think, right? Like, we think it's a lot of noise. So this is a really, really, I will say, outstanding result that variance is actually very well conserved across studies.
Isn't this from your design by removing the tissue-specific expression that you get exactly rid of those ones that will be more heterogeneous? You go more for the processes that are all changing.
No, there's a lot of action in 65% of the genome. But of course, the ones you cannot find in one tissue, you cannot study because it goes from variance 1 to 0. Okay. It's because, yeah, absolutely. You are biased to things that you can see, and they have to be biased by things that are always needed in a cell.
It's a huge variation, right? I mean,
Huge.
big neurons are doing a huge amount of all the time.
Exactly.
Some cells
And the levels of gene expression, yeah.
So
Yeah.
even for housekeeping genes, you can still find variation in expression. That's the main variation. And so even for housekeeping,
This is variant across people. Especially this variant.
This is
This is variant across. I'm not sure. Variant across what?
This is, you estimate variance in sample 1 of gene A. And then you estimate variance of gene A in sample 2. And variance of gene A.
People.
Yes, exactly. So this is your 400 samples in experiment 1, had 400 individuals.
1, 2, 3, 4, 5 extra variants.
So, but 1, 2, 3, 4, 5, what?
Individuals. Okay.
The variance come from, exactly, from your sample size in one particular study. That could be 400, could be 20, or could be 1,000. So you estimate variance there. Yes, all these individuals are, are humans. They are, you also have that there. So this is, you want to imagine this, is going back to the universal dramatic statements I make, right? Like you have genetic variance, right? Like this is an average of individuals, a variance of individuals. You are put in different environments, a technical noise that is there. Different environments, certainly, because we don't know what's going on there. And does something remain after all this stuff is kind of the question. Good. I'm not gonna try to, yes? Too long about this, with this, but once you know variance is real, we believe it, we know it's gene specific, then you can go and ask evolutionary questions, like what are actually the forces that make this variance remain certain way or the other. I'm not gonna go in detail because of time, but you can find, that genes that are more variable are more diverse, genes that are more variable, less variable are more intolerant to loss of function, so they are very sensitive, you cannot mess up with them. They are highly connected in gene networks, and this is super interesting, right? Like if you are reliable, you have many friends, they can rely on you, you are always constant. If you are one of these guys that you don't know what's going on, it could be here, it could be there, it could be in the middle, you're poorly connected. My comparison is always that you cannot rely on these people that you don't know actually where they stand, right? And this is true for gene expression. Functional enrichment. This is also very interesting because the enrichments are really, this is always what we want as experimental biologists to find, right? Is there some biology behind our stats? In terms of geo terms, usually. So you see what is maybe not surprising. Immune response it has a strong enrichment in genes that are highly variable. And we evolutionarily might want to keep these genes in certain varying a lot. And you have a lot of housekeeping functions being these genes that are really tightly regulated, which again also makes sense. Good. So let's say that we agree that variance matters, right? And now the question is, maybe going back to what Julia was mentioning, now what about the environment? I'm telling you the whole talk that the environment is modifying all this stuff. So how do we measure that? So again, we go to flies, because we can actually do experiments there. And this is the work of Jain. He's a PhD student in the lab. And what we are doing there is doing our usual trick of outbred populations that we know are very good for mapping. We are collecting around 1,000 samples of RNA-seq and DNA data in control on our favorite, high sugar food. And then we end up with around 2,000 matching DNA RNA-seq pairs, right? So you can actually do powerful, statistically speaking, EQTL mapping in the samples. And how do we do that is, again, one of the comments I made the other day. The questions you can ask only depend on which methods are available for you to collect data when you are an experimentalist, right? So when I started this project, one sample of RNA-seq was like $30 or 40. Like you needed to buy the kit to make these particular libraries, which means, therefore, the sample sizes of all these experiments are very small, right? And we cannot do EQTL mapping G by E, right? Like environment dependent, if you have 100 samples, right? You're never gonna, it doesn't work. So we put a lot of effort into developing a method that actually allows us to make RNA-seq samples. Back then, this is already a while ago, was for, I don't know, like $2 back then, now probably because inflation. But $2 from 40, right? So we actually do all this stuff we do in the lab homemade, right? This is based on TM5. If you guys make libraries, you know. All this is as much homemade as you can, so you don't have to buy anything from kits or anything. And then when the library goes down to €2 to $2 or whatever, then you can do it, right? Just to highlight again, many of the questions we want to answer, we couldn't just because we just couldn't collect the data. This is just, I'm not going to talk about that, but this is just to link it. I'm trying to link it to Bruce's talk that was mostly about the UQTL mapping stuff. So this is just a paper that we put together mostly as a resource. We are trying to do, what all these GTech catalogs in humans do, in a sense that people who are mapping stuff in Drosophila, they can go back to a EQTL map and try to see if EQTLs for traits actually are explained by EQTLs in expression. And this is just to show something that is really sad, or really, I don't know how to interpret this. This is the number of genes with cis-EQTLs that you can map. And this is the number of individuals that you use. So this is our data set. We have 1,000 samples for the body and 1,000 samples for the head in flies. This is as good as a tissue that we can get, again, making it feasible experimentally. So in total, let's say 2,000 samples. And you can see what you get with 200 samples. You get around 20% of the genes expressed in these tissues in the fly. And this is how much you gain when you add 200 more samples, and et cetera, and et cetera. I just want to highlight here that with 2,000 samples, this doesn't even look like it's going to plateau ever, right? And I say sad in the sense that we know this is super complex threads. You have this really, really long tail of small effects. Do we want to go into more than 2,000? I don't really know, right? Like, do we want to, what's the point? But this is just to highlight how hard it is really to map these really small effects in, for example, gene expression. And I just want to highlight that this little arrow here, intentionally made very small, is the number of cis-EQTLs that we had for flies until last year, I think, right? So this is what was done before. Again, huge efforts, right? Coming mostly from inbred lines, but everything goes down to power, right? If inbred lines are 120 or something, this is not a surprise that the power to detect stuff is lower. This doesn't matter. This was just to show you a real EQTL map, going back just because Bruce mentioned it. But if you are thinking about EQTL, just talk to me later. Good, in the final 10 minutes, I'm gonna try to tell you this particular story about gene expression variance in flies, depending on the environment. So this is just for contrast, each dot is one of my 2,000 flies. This is the transcriptome of one fly. And this is a principal component analysis showing you high sugar and control.
Sorry, I forgot.
Fly.
Where are the 2,000 flies sampled?
Where?
How are they sampled?
They are, again, these cages of populations that are exposed to two diets. So it's the same genetic pool in two conditions. So this is just to show you, sure, gene expression, transcriptomics, the profile changes with diet. And this is just, if anyone has done differential expression analysis here, this is what a volcano plot is. These are all the genes that are differentially expressed using the mean, right? They have a significant change in mean expression. This is what we all do when we do gene expression analysis, right? These are the genes upregulated in high sugar, downregulated in high sugar. I could give you a whole story, a whole talk about what happens with mean changes. But I'm just bringing this here as a reference, because now we're going to forget about the mean and just focus on the variance of these particular genes. So this is how it looks for mean. This is how it looks for variance. So gene expression variance mostly increases in the new environment. This is, again, the same thing. You measure variance based on the 1,000 individuals you measure in control, variance on the 1,000 individuals you measure in high sugar. Do they differ, right? Again, all this is corrected for the mean. We don't want the mean to affect anything we do in variance, because in gene expression, these two things are always coming together. And this is the same. Of course, it cannot be called a volcano plot anymore. Anything that goes to the right is increase in variance in high sugar, decrease in variance in control. You can see that this is really a strong trend of when you put these little flies on high sugar, variance in gene expression explodes. This is just to show you the colors here is that 47% of them are accompanied by mean changes. But I find fascinating that half of them only change variance. So if you remember the cartoon I showed you before, these are means that stay the same and variance that explodes. Good. So what is it behind this? Like, can we? Yes.
6% that only variance?
No, so you have 82% of the genes in the genome have different variants. Half of them have also change in mean. These are the genes that were differentially expressed in mean.
Half of that 80.
Correct,
Okay, yeah,
correct. And half of them in variants, yes. I will tell you in a second. So the first approximation for us is what does it mean? Can I learn biology from changes in variants that are not changes in mean, basically, right? So we try to dissect this. This is still like very preliminary. Is this the best way of analyzing this? I don't know, you let me know. So what we did is, okay, we take this is control and high sugar conditions. This is the variability rank, right? This is the most variable gene. This is the least variable gene, right? So if we take the top most, these are 450 genes that are the top most variable genes in control. You can see that most of them remain highly variable in high sugar, right? So genes that we started already with a lot of variance actually cannot move more than stay with a lot of variance. Ranks, we can talk about the absolute increase in variance. This is, again, for contrast, what happens in mean. In mean also, genes that are the top expressed remain the top expressed.
The variance is normalized by the mean or by the standard deviation normalized by the mean?
Here we are using MAD, so median absolute deviation, to estimate variance. So we are not using SD or any other property to measure variance, just because gene expression has also this property of weird, funny outliers that drive all your estimations. So when I say variance is MAD, so median absolute deviation, but before that, we have normalized the data to remove the effect of variance. So if you were to plot mean expression and variance, you had a flat line. This is done before.
But I guess I'm wondering, how is it normalized relative to mean? I mean, some of these where it's 10% variance, 10% variation around the mean, or
So you say the coefficient variation of species is constant, so log transformation.
I see. So you're saying so that's not.
That gives you the baseline signal.
Right.
Good. So you have two points, right? So first, you have an intrinsic relationship between mean and variance. This is a property of gene expression. Things that are really highly expressed are going to come with high variance. Right. So before you do any analysis, you have to make sure that this is not there, right? So you have to adjust the relationship between variance and mean to be flat. Otherwise, everything I'm telling you here is a mean change. So this I couldn't tell you mathematically. So the method is called variance stabilizing transformation, and this is something that people develop for a long time to actually, when every gene expression analysis that you see has this variance stabilizing transformation.
But this work where if you want to track cell lineages, and you look at transcription factors, often you look at transcription factors and you can figure out which ones are important by looking for the ones with low grade. So there are, now maybe they are negligible.
It changes.
It doesn't matter. There are definitely ones with consistently low variance in some maybe by the time you mix together a whole tissue, that's no longer true. But I guess the tissue I know is blood. Some people get their tissue don't. But it's then looking at these different cell types that have lineage. But I guess it's a pretty small fraction of genes.
That would be that.
Most of them, by the time you log transform, and they look.
Yeah.
So another way to frame it really simply, if you have some genotypes that have higher variance expression for a specific transcript, you can have some map steps for that.
So this is just to tell you that because they remain highly variable, it's no surprise that the geo-enrichments that we see is actually response to pheromones, response to environment, sensory perception of smell, like all these things that make sense that you have to have them there to perceive your environment and react to it are highly variable. And this is, I like it because it really seems something that has to remain this way, regardless of your environment, right? Uh-huh. Uh-huh. Because they couldn't sense that? Oh, I didn't know that. Exactly, to perceive what's going on, yeah. And now, the bottom part of this distribution, right? So this is really interesting because then you get, again, for reference, genes that are lowly expressed, they remain lowly expressed, the mean plot. But when you go to variance, you have genes that are at the bottom, they don't vary in control. There's this explosion of variance when you move to high sugar. And the question is, what's the biology? Why am I exploding variance there? So here, you have 50% of the genes actually remain. So there's a core also part of genes that, sorry again for this wording, need to remain low variance in these conditions. And in flies, it happens to be reproduction, right? But this we know, right? Like these flies are fantastic doing quality control when they are making eggs. They might lay different number of eggs, but the quality control in producing eggs is really, really tightly regulated process. They kill crappy eggs in the middle when you put them in high sugar, and the ones that come out are actually as good as the control ones. So this was really interesting. The things that need to remain constant is associated with reproduction and egg production.
Are those also more constant?
Oh, I don't know, in flies? This is I don't know if we have good resolution for tissue gene expression. We have atlases, but I don't know if you could actually measure variance there. Yeah.
Still, maybe if these are mostly housekeeping genes,
then would remain. Maybe it doesn't really matter. Yeah, could be. And the cool thing is now, this is actually not the best plot. There are 47% of these little genes that are low variance only in high sugar. So I told you that, like most of the things increase. I should have plotted differently and show you the ones that are here that actually come here. They are, again, don't take this word too seriously. They are canalized. Their expression is canalized in the new diet, which is fascinating given that in general they increase, right? What are those genes that need to be tightly regulated in the new environment? They are all enriched for metamorphosis processes, which is very interesting because when you measure flies in high sugar, their development is really delayed by 30% or so. So developmental processes that are bringing you from the egg to the adult are actually, we know, different in high sugar. But somehow it seems that they need, even is lower, or maybe because they are slower, their regulation needs to be tighter. Yes? This is one generation. Yes. Yeah. Correct. Mm-hmm. This is one of the main questions we are tackling in the lab. This is what happens if I freak you out now, right? Is this adaptive? Are these changes any relevant at all in evolution? Maybe not. So we do experimental evolution. Correct. We do experimental evolution with these flies and ask me in a year or two, and then I will tell you, are all these things that we thought were relevant for example, increasing the range of phenotypes you can reach, are they actually relevant for adaptation to high sugar? So we are now in the 25th or so generation. So this is running, and I will be able to tell you all that. Next, for the next workshop.
The question was a different one,
Oh.
not long-term evolution.
Oh, okay.
You haven't done a common garden, if I understand it correct. So you've just done what we call a sugar shock. So how
Yes.
does it behave if you could have done a common garden?
What do you mean a common garden?
Well, that they have been at high sugar for 2 generations.
Yeah.
No adaptation.
That's what I'm saying. But only for 2? Not for too long. Oh, I don't know. Because what we are doing is just assuming that at some point they will adapt to high sugar to evaluate whether they are adaptive or not. But not really short.
It's more
Yeah,
like
yeah.
French generation effect.
Effect,
and
yeah,
the
yeah.
diet, I think, is particularly important.
Yeah. We haven't, and I don't think we will do, we will do it in terms of the amount of data that you will need. But yeah, it's a good point. Okay, I need to let you go. So this is just to show you same patterns that we found for things in flies that were very similar to humans, except for connectivity in gene networks. And I'm gonna leave that so in case somebody has an idea. I told you this little funny, beautiful story of low variance being very reliable genes that have a lot of friends in the gene networks, right? And unreliable genes have little partners in the gene regulatory networks because they are unreliable. And I hope you believe me. Okay, this is humans. But now if you check data in flies, we find this beautiful smile. When you have, if you are not so variable, you have a lot of friends, this follows my narrative. This makes sense. But when you get to the tail of this distribution, genes that are really the top more variable genes also are very well connected. So all my story of friends, reliability, no, gone. It doesn't work here. Why would you be very well connected in the extremes of this distribution of variance? I have no explanation for it. And I'm bringing it here so you can think about it. Please let me know. This is how many genes you are connected to.
I'm just trying to extract the power law or whatever the connectivity is.
So this is the number of genes one gene in this class is connected to. So if you estimate co-expression networks, how many of these genes have partners, basically?
Wow, so you don't have the power law.
Correct, but if you see it, the thing is like, for example, our results in humans show that, but it's really like, it's not like a beauty, it's really like borderline significant. Its effect sizes are very small. So it could be that it's also true for other systems, just that the data probably is, I don't know, or it might be that the regulatory networks in flies are different.
What is the expression coming?
Here is co-expression between two genes. So you estimate correlation between expression levels of two genes. We adjust all that super strongly for full discovery rates and stuff, because you get
Covariance.
correct
Being
covariance of every pair's way
looking at the variability and the variance
across in the whole transcriptome, and then you count how many partners do I have. So how many genes do I covariate significantly with? Yes. Mm-hmm. But hab genes tend to be here. The variance is way smaller than non-hab genes. That's when this narrative breaks, because this will tell you that non-hab genes, so if you want to think in terms of an omnigenic model, all your peripheral genes will be the more variable genes, less connected. But somehow here, we seem to have this, like, and I don't know really what's going on there. Okay, I'm over time now, and I still have a lot of things to tell you, but I won't. I just gonna finish with this little picture. Oh, Bruce, we have to talk because we have fluctuating asymmetry for our diet.
So it's higher in the high sugar diet?
No.
Lower?
Are you familiar with this cross-vein Waddington vein in flies, which is a proxy for developmental robustness too? We find the opposite. High sugar makes them more developmentally robust. But I can tell you stories about that later. I just wanted to finish with Bruce's picture. If you recognize it from the class 2 days ago, and he was telling us all the things you can map, EQTLs for everything. And I was very happy because the last one was the variance EQTL. So all this story I'm telling you so far with the lab you started, we are approaching what happened to phenotypes when you are in a new environment. What happens to variance to phenotypes? But the goal is, are these things genetically regulated? And if so, what are the pathways that regulate variance control? We don't know. So a lot of what we are doing, therefore collecting all these samples, is to be able to map variance EQTLs, which at the end is this flavor, when your genotype doesn't change your mean, but changes your variance. More on that in the next workshop. Yes, Julius. Mm-hmm. Mm. Yeah. That increases together for all of them. Yeah. Yeah, we could check that. Yeah. Yeah, that's a good point. Yeah. Thanks. Yeah. But then you would oh yeah, no, that's a good point, because you wouldn't expect it from the middle, because these guys maybe don't change invariance so much. Yeah, yeah.
So since we're running on the coffee break, let's thank our speakers.
So sorry to take your to go over time. Thank you.